So DiabInnov is here to help you to fill the gap between your experimental work and your clinical developments. So for that, we will propose you 3 original tools that have been developed during the 2 last decades in our team in the academic setting of EGID and our INSERM team to propose new and innovative therapies for the most severe forms of diabetes, including cell therapy for type 1 diabetes and metabolic surgery for type 2 diabetes. So DiabInnov was developed as a front office for our unique academic setting. Our team indeed is devoted for now 2 decades to the development of new and innovative therapies for type 1 diabetes, but now type 2 diabetes, obesity and other metabolic diseases such as NASH. Our team is gathering together clinicians, both surgeons, endocrinologists, hepatologists, and cell and molecular biologists, all together working to help you to answer your research question. One key asset of our platform is to be coupled with one of the most active islet transplantation programs worldwide.
So human islet production takes place on the biotherapy platform, a French FDA authorized facility. Over 1,000 pancreases have been processed. So working with human islets is an essential step towards bridging the translational gap between murine studies and the clinics.
This will give us access to islets from numerous donors of different type and different phenotype, and considering the heterogeneity of the human species, islet speaking, this is a major advantage when you will address specific questions in the human species, to be able to work with our tools for dynamic testing of insulin or other hormone secretion, both in vitro with perfusion or in vivo following transplantation in immunocompromised mice.
At DiabInnov, the unique quantitative in vivo mouse model with a human islet graft can be equated to a phase 0 preclinical trial or test to screen drugs or molecules to validate their effect on human islets in vivo, and in particular on islet function, proliferation, mass, gene expression. DiabInnov also proposes 2 other variants of this model, where mice are grafted with human islets and we put them on high-fat diet or render them pregnant, and DiabInnov tests the capacity of different drugs to rescue the dysfunctional phenotype.
DiabInnov has a full access to our state-of-the-art large animal facility at the crossroads of the research teams of the University Hospital of Lille and INSERM, including a large capacity animal facility for small and large mammals and operating theaters.
So DiabInnov offers a great opportunity to work with large animal models. This kind of models, like the porcine model, give a great access for surgical and endoscopic procedures.
It is eligible for numerous regional and national calls to support projects involving academic and private partners.
The process, from the idea to the market, has many steps to fulfill. All of them, from ex vivo industrial experiments to preclinical and clinical studies, to establish safety and efficiency of all devices.
So working with our cohort, the ABOS cohort, a unique setting which has now enrolled more than 2,000 patients, all with severe obesity, candidate for bariatric surgery, which have been submitted to deep phenotyping both before and after surgery, with a 5 years follow-up for most of them, as well as tissue and other fluid samples, both at the time of surgery and during follow-up. And that includes tissues such as liver, muscle, subcutaneous or visceral fat, all together for the same patients.
DiabInnov promotes dialogue between industry, scientists and surgeons to facilitate CE marking and patent obtention.
Based on our previous experience in developing medical device and innovative therapies in type 1 diabetes, we will help you through interactions to formulate your questions, elaborate the models we'll work on together, and answer the best way we can the question you're raising. So in summary, DiabInnov will help you to accelerate your project from the clinical experimental data to the clinical development.